Rosa told me about it. You should come.
Yeah, definitely. Okay. Do you know where it's at? Is it in, yeah.
Pretty sure it's in 209.
Okay.
They had it started at that moment. I hope it's already done by then.
Alright, just so you guys are aware, I already hit the record button just to make sure I don't forget to. So your conversations are no longer completely confidential, but since I've failed to hit record so many times, I just started early today.
Thank you for letting us know.
So you're just involved with talking about it?
Actually, no.
Other people water and food, so,
I tried to find out really excel. I might go back to it. I applied for it and was offered it for 6 months. And so that was offered in the year. Well, still maintaining a certain core of
okay, yeah. That's too much, yeah. Well, it's just Kevin has it rolling still, and he has some, elders that are.
okay.
Just, I mean, from my interviews, people helping out. Yeah. I don't know what his thing is.
Yeah, so he loves doing it, so I know it's sometimes challenging.
Yeah.
Find a way to get it done.
Yeah, well, yeah, I'm not, how, I mean, he's got a lot of people there.
Yeah.
It's pretty impressive stuff.
You know I hit record, Mary.
Record.
Yeah, so
Like
all
our
conversations are being recorded.
I appreciate you letting me know that.
Just getting the heads up.
That may in fact change my behavior.
It usually impacts mine positively. Yep. Yeah. We'll talk about them during the presentation, but okay. And then we have these fact cards that our team at UConn made, so if you guys want to take one, many, I've got more over there if you want to take some for those of you who have farm stores or farm stands. Just for some basic information, you're welcome to these too. So. Alright, well, yeah.
I'm Suresh, I work here.
Okay, oh, you, okay.
Yeah.
Okay, yeah, I remember you got that, you were,
Yeah.
Do you look into this?
He's not wasn't my advisor, but
Oh, okay.
I'm involved
With
some
some of the
projects.
productions. Okay, yeah. Nice, yeah. Yeah.
How do you know this?
So, I was going to be there, I was PhD in science here. And so, she's not a good.
How was yesterday at the event?
I didn't work directly there, but yeah, she grew up.
Yeah.
Yeah, small world.
Yeah. Are you going to this year's conference in Las Vegas?
I'm not planning on it. We're going to cultivate, it's more of a nursery expo deal. And then usually I go to plant animal meetings. San Diego, so yeah, I'm coming back. How are you? Yeah, oh, okay.
And then Lisa will be there too.
Yeah. Las Vegas in July.
Yeah. Even around 100 degrees or so.
Yeah. But there should be a good turnout there. Like, that's it. Okay.
Season.
Yeah.
Or no, stores.
Oh, you're just in South, okay.
Some land here that's not being used. No, it's
Oh, yeah, December is supposed to come too. Hello. Oh, you're the holder. Okay. Hi. Thank you for coming. Yeah, Mary Conklin said you were coming, so it's like, oh, we better not start it up then. That's me. Okay.
I'm waiting for one more from our client.
Okay, we'll wait. Hello. There's snacks and bottled water in the kitchen. Help yourself to whatever you want.
Thank you.
Oh, yeah, I have more for you too. I'll get them out. Yeah, no, I told you, And this one is the shortened version of that one, right?
You know, that's, yeah, that's definitely fine.
Okay. Don't worry, I'll do it. There is a lot.
Yeah, we can get started.
Well, good evening, everyone, and welcome to the Town Extension Center. I recognize a number of you folks that are around the table, and some of you I don't recognize. We're going to go around and introduce ourselves in a few seconds. A couple of logistics. Obviously, we mentioned that we really have some water and cookies and some fruit and some granola bars and some chips and what have you, so please feel free to help yourself to the refreshments. I already asked about coffee, and apparently I have no coffee drinkers here this evening, so I'm not going to make coffee. I hate to throw it away if no one's going to drink it. So, but again, if anyone changes their mind, I'd be more than happy. It's all ready to go, so I'll be happy to do that. Bathrooms are right down the hall to the right for both the men's and women's rooms. So feel free to do that as well too. Before we get going with our guest, I'd like to get stuff around the room, go around the room, and kind of introduce yourself, maybe a little bit about your business, if you're a farmer or how, what you do.
I'm Mary Ann, and I'm just starting an organic farm in New Jersey, actually. But I live up here. It's family name down here.
Go ahead.
But so I'm having mostly perennial crops. I would like to use some of, I guess GMO, the cottonseed meal is a good fertilizer for food areas. But I put that question out in the application when I signed up. But I can't ever seem to locate the non-GMO because we're not allowed to organic to use
GMO.
Yeah. So I guess I've had a couple questions people asking about GMO, so that's why I'm here tonight.
My name's Amanda. I'm a dairy farmer in the northwest corner of the state. So we milk about 300 cows and grow about 300 acres of Roundup Ready corn. We also manufacture a biodegradable pot with our cows in our called cow pot. So I actually interface a lot with the public when it comes to the sale of that product, and quite often I'm asked about what our cows are fed because I'm concerned about being able to use our cow pot. So I have the response that I've generated, but I'm eager to see how I can adjust that response.
Okay.
Carol Say from East Dickens Orchard at Menfield. Both of us are, and it's fruit, vegetables, berries, and we've got a pretty wide exposure to the public because we're doing pick your own, we're doing farmers markets, we're doing CSA, and we've spent a good amount of time the last several years massaging the conversation around IPM and organic and sustainable and that kind of stuff. We're just barely starting to get questions on GMO and need as good a conversation on that.
Mm-hmm. Okay.
I'm one of the farm managers.
I wrote them.
I'm Katie Urbana. My sister Ashley and I lead Hudson Turkey Farm in Sterling, Connecticut, Eastern Connecticut. We raise poultry and livestock. Our livestock is grass-fed, but our poultry is not. We're buying in our grain, obviously. And we've had a lot of questions now that there's an availability for GMO feed and bulk in the area. We question whether we should go that way, but as a family, we don't want to succumb to the pressures. We support GMOs. So we just want a better way to talk to our customers about it.
I'm Diana Holder. I'm from Holder Project in Southeast Corner, and we don't get an awful lot of questions from customers, but occasionally we get some questions from customers. Right now, we do program optimization on farms and looking for a better way to either express things or better ideas.
Just introducing yourself.
Hi, I'm Bob Alex. I'm from Watertown, various investors, small
Dick of Bottle Farm in Ashford. We're a non-GMO farm, sustainable agriculture, all doing livestock. Great idea. So, the other side.
All right, great.
Hi, I'm Suresh. Vegetable extension educator. I work in this office. Work with commercial vegetable growers in the state.
My name is John Mahoney. I'm a PhD student at UConn studying plant breeding, and I work with a small fruit crop called Aronia or chokeberry. So I don't know if any of you are familiar with that, but we can always talk about that later too if you're interested.
Kevin Woolen, used to be in the feed business with our co-op. There's some feed on the side still and livestock producing.
Okay.
My name's Rich Coles. I'm an entomologist at the Connecticut Agricultural Experiment Station.
Excellent.
And I also have a small farm with growing Christmas trees in Broadbrook. And expect to be hiring a postdoc to be genetically modifying horticultural crops for resistance to various pests.
Oh, okay. Very good. You I'd like to talk to.
Okay.
I'm a plant pathologist, so there's a lot of...
Well, I'd like to also introduce you to Dr. Vincelli, but just maybe a little bit about his credentials and where he hails from. Dr. Vincelli is an extension professor and provost distinguished service professor at the University of Kentucky. His work focuses on plant pathology, including management of diseases of turfgrass and forest trees, molecular diagnostics, international agriculture, sustainability of food systems, and outreach on the risks and benefits of genetically engineered crops. Paul serves as the co-coordinator for the Southern Region Sustainable Agriculture Research and Extension, SARE program. He earned a Master of Science in Plant Pathology from Rutgers and his PhD in Plant Pathology from Yale University. Paul served as a botanist in the U.S. Peace Corps, as a service member in Colombia and Nicaragua, and a Fulbright scholar to the Uruguay and Nicaragua. So again, we're pleased certainly to have him here, and looking forward to his presentation this evening.
Well, thank you, Joe. It's a pleasure to be here and interact with extension groups from here in Connecticut. So really, that was very helpful, Joe, to go around the room and hear sort of the different perspectives of people. The presentation itself is largely focused on how to talk about genetically engineered crops, and it does come from my bias or judgment, whichever you prefer to call it, of and my bias in this topic, I don't know, again, correct to call it bias, is that genetic engineering provides a set of useful tools. Doesn't mean everybody should agree to eat GMO foods. I'm not here to argue that, but that I want you to know where I'm coming from. So this presentation comes from that perspective. In addition, I think some of you asked or presented yourselves as interested in particular aspects of GMOs, and so I want to make sure you know, if that's what you came for, to please make sure you ask, you know, the questions that you have at any point in the presentation if they come up. Okay, so, but I feel like I should focus on the presentation because there's a lot there, and so how about if we do that? Okay. So thanks for coming. Appreciate it. Oh, you're all right. Yeah. So anyway, anyway, so let's get started. I'll just use the down arrows. Those. All right. So there are two major topics here. One is ways to communicate the key biology of genetically engineered crops. And so that'll be of interest to some of you. And the second is guidance from social workers or social scientists. My wife is a social worker, full of good ideas that are born out in the academic literature. So she's really been very helpful. But this whole section doesn't just come from her; it comes from a lot of sources like her, and it's really helpful, actually, no matter what the controversial topic is, to use some of these principles. So we're going to go into that too. But we'll start with the ways to communicate the biology of genetic engineering. First of all, genetic engineering is already part of our lives in ways that we may not know. And like if you know a diabetic, their insulin is very, very, very likely to be produced by genetically engineered bacteria. And as a result, it actually is higher quality than what you would get from the pancreas of animals that have been slaughtered. So that's a common application. There are others. All right. So again, this is about how to talk about genetic engineering. So this is a mix I made myself of dried cranberry, and peanuts, and it's all non-GMO. There are no GMO peanuts. By the way, I say GMO, but actually most scientists prefer the phrase genetic engineering because it's a more precise way, as you will see, of describing this. But anyway, so I might say for shorthand GMO, but this is non-genetically engineered, so it's non-GMO. And so the question is, am I eating DNA? If you think I'm eating DNA, raise your hand. All right, good. Some hands going up. All right, good. So this is important because you know, it helps make the point that DNA is a normal part of our diet. Okay. And I say this, all right, so if I eat a banana, will I become a banana? I'm glad you're finding this amusing because it's meant to be. And the answer, of course, is no. But it's important that when I give talks to the public, it's important that people understand that DNA is a part of our diet and that it's not harmful, okay, to eat DNA. So this is corn. You recognize that. This is the progenitor of corn. And through human selection, that became, which is called teosinte, became this. That wild grass became this. And so were genetic modifications, did genetic modifications occur as a result of that process? And by genetic modifications, I mean any genetic change, not just in the lab. Of course. Yeah, right. So that's another way to help people understand that genetic modification itself is not harmful. All right, so take a look at this. Again, these are all sort of introductory concepts that I hope are useful. But these are portions of 2 chromosomes from tomato. So chromosome 10, a portion of chromosome 10 is represented here, and a portion of chromosome 7 is represented here. And those arrows are genes. And so that's a pretty big piece of chromosome 10, but of course chromosome 10 would wrap around the room if we plotted the whole thing. But anyway, it gives you a pretty big chunk. And so this, look at what happened. Okay? So that, a big portion, 24,000 DNA letters were duplicated and inserted into chromosome 7. And notice what happened to chromosome 7, that particular gene gets split apart and becomes non-functional because of the insertion. Okay, so you could look at that and be puzzled by it. You might have some particular thoughts about it. What it is actually a natural duplication that occurred that led to the production, that produced these Roma tomatoes, which we use in tomato sauce and the others. So genetic change is part and parcel of the world we live in, okay, and the food we eat. Have you heard about this case?
Oh, yeah.
Yeah. So sweet potato is actually a natural GMO, and really all foods are, but sweet potato is the most recent example that I know about in the scientific literature. And so what was reported back in 2015 that at least 4 bacterial genes are present in the genome, in the genetic biology of the sweet potato, of cultivated sweet potato. At least 4 and as many as 9. So really, you could think of sweet potato as a natural GMO.
What functions do they have?
I don't think we know yet. My assumption, this is just a sort of an assumption, but because they're in all cultivated varieties that have been tested over 200, it must have conferred an advantage, probably in size, I would guess, back the thousands of years ago when this was first selected for propagation. But we don't know exactly at this point. We do know that those genes are expressed, at least some of them, so they actually are functional. This, and if you interact with the public, this is a peer-reviewed. I've talked to genomics experts. This is a peer-reviewed way that you can communicate about what genetic engineering is, and that is to use the word processor idea. So let me show you what I mean. So imagine that I'm of Italian descent. So if I write a cookbook, it might be about Italian cooking, right? So with some of my mother's recipes as best I can remember them and so on. So imagine I'm writing a book on Italian cooking, and I want this particular sentence in my book, and from one file into this book. Okay, and so we think of the sentence as the gene and the file as the plant. Okay, and so in the older techniques that are still used and they're still useful, but the older techniques of genetic engineering are called plant transformation, and it's basically a copy and paste mechanism. And so like you might do with a computer file, you copy the sentence and you paste it into another file. Okay, and so the weakness of plant transformation is that it's a random insertion. Okay, so that means that some of these insertions. Okay, here we have the insertion. So there's the sentence. There's the sentence, and it's fully present, which we can test in the lab, and if the gene's there and functional. And if it is, then we've accomplished the insertion. But maybe the insertion took place in a place that could be harmful to the plant. So that's why we would test maybe hundreds of these and find a few that we want to carry forward for future analyses and work. The random insertion, I mentioned that, but you can always overcome that by just doing enough numbers. Okay, so now I'm going to talk about the latest techniques of genetic engineering called CRISPR, or gene editing. How many people have heard of CRISPR? Okay, most of you have. Yeah, this is a well-read group. So this is to me, this is one of the most important slides or a series in this presentation, is to at least understand this technology. So, and it's about making targeted edits, which is different from copy and paste. It's actually more of a targeted edit phenomenon in the biochemistry of what actually happens. So we've got this sentence in the introduction, let's say, of my book. The precursor of pizza was probably the focaccia, flatbread known to the Romans as panis focaccius. You say, you know, that really could be improved by inserting that sentence right here. So you can't prearrange that insertion with plant transformation, but you can with CRISPR. It's like typing it in. Okay. So that's the level of control that is now available to biologists, and it's why there's a lot of excitement in the world of biology over this technique called CRISPR. So let's say a little bit more about that. Again, it's targeted edit. So let's say, you know, clearly wine is not made from grape juice, it's made from grape juice. So there's a genetic defect, right, in that gene, let's say. And so it's missing a particular letter that it needs. And you can in many cases, insert the letter that you want, and more cases in more cases every year. So you can basically make a genetic correction like that. And there's a lot of interest in sickle cell anemia for that reason, for people who suffer from a genetic defect that could be potentially corrected by CRISPR. So the genetic defect might be wine is made from grape juice, so we want to get rid of that particular letter, and we can do that with CRISPR. So why would we want to do that, at least in the plant? Well, so these are basically identical, nearly identical fragments of a particular gene in beef. It's a particular gene that was selected for this reason that you'll see in just a minute. And so the wild type sequence is given on the top, and the bottom two represent plants that were CRISPR edited to result in what we call a small knockout. Okay, so a few nucleotides, a few letters were knocked out as few as one, and the genes suffered a loss of function. And so, again, why would we do that? Well, first of all, this is just to illustrate that that is an extremely modest change. I mean, as minimal a change as you can make in the genome of that plant. And this is why they did it. So the upper leaf is covered with powdery mildew, which is a disease that our growers in Kentucky spray for sometimes. And the lower leaf is one of the leaves from plants treated in this way. So actually, by knocking this gene out, and they anticipated this. They didn't just do it blindly. They knew what the gene did. They anticipated that by knocking the gene out, they would create a resistant plant. And so these are this particular type of application, small knockouts for a change of function, there's a lot going on in this field, in all kinds of fields, human biology, animal biology, and certainly plant biology. So to contrast that, and feel free if there's something you want to drill down into a little bit more, just let me know. But I want to contrast these techniques which have strengths and weaknesses, okay, to the non-GMO breeding techniques, which also have strengths and weaknesses. And just for comparison, so let me give you just a few examples. Maybe, I don't know how many I've put in here. Yeah, I put in 2 or 3. So hybridization, we all know something about that, and we're comfortable with that technology. And I'm going to use cards to illustrate this, okay? Again, this is something scientists have had the chance to comment on, and it seems to be a well-accepted analogy. So imagine that this is a plant that grows well here in Connecticut, okay? And it survives the winter very well. It tastes good. It withstands the hot summers, whatever else. But it doesn't have resistance to a particular disease. This is a wild relative of that, and it has, by the way, the cards are the genes. And so, it's not going to perform well in Connecticut. It's going to taste bad. It's going to grow slowly, poor yields. But it happens to have this gene 5 times that confers resistance to the particular disease. So hybridization is essentially, on a basic level, that. Okay. So what we've done is we've caused a lot of genetic change by mixing these two. Okay. And you can reduce the presence of the blue genes through backcrossing. I could explain that more if you want to go during the Q&A. We can talk about that. So, but we'll never get rid of all the blue genes. Yeah, all the blue genes. So we've permanently changed the genetics of the red parent. With genetic engineering, the idea is simply you take the gene from the source of interest and you insert it into the plant. And that's the basic concept. So hybridization actually causes more genetic change to the plant than does, quite a bit more actually, than does genetic engineering. Oops. So here's one that usually gets people's attention. It is possible in the lab to strip away the cell wall of a plant cell. And then they, in buffer, they will sort of turn into these round balloony structures, and those are plant cells. And you can actually fuse these together and basically smash them together and force them to hybridize, usually within the same family, though as far away as the same family, but sometimes even further. Plants that never would have hybridized in nature. And that's a non-GMO technique. That's actually a non-GMO technique. Here's one, again, that's even more surprising. People sometimes ask, are you sure that's non-GMO? It is. It is a non-GMO technique. So this guy is protected from this very potent mutagen. EMS is the abbreviation for it. And why is he doing that? He goes out into the field, and as part of their breeding program, and they dab this highly mutagenic chemical onto the silks. Pollen lands on the silks. It gets highly mutagenized. Many of the pollen grains die, and so, but some survive, and they've been mutated in ways that may be harmful to the plant. But a few of them may have some useful mutations that can be propagated and used in the breeding program. It's called mutation breeding, and clearly that is a much more disruptive technique than inserting a couple of genes through genetic engineering. All right, so I've already answered this question. Which causes greater genetic change, genetic engineering, or conventional breeding? Conventional breeding does. I've already done that. So I've given a number of slides here in various ways to illustrate, first of all, that genetic change is normal and desirable. If we lived in a world without genetic change, it would be a poorer world. Although genetic change can be harmful too, right? If you have a mutation that causes cancer. So, but genetic change in our foods is normal and desirable, and it comes about in lots and lots of ways. So so this is another principle. Again, this is not me talking. I'm representing to you peer-reviewed science. This is all easily documented, and you should all have two handouts. Probably the one that is most important for you is this one on consumption of genetically engineered GMO crops. These are, I'm going to come back to this publication actually, but they represent major scientific societies throughout the world. And so you can read some of the very same things that I'm saying, in these massive and very, very credible summaries. So genetic engineering offers more control when making genetic change. And again, this is scientific societies have been saying this for decades. With respect to genetic change, what matters is not how the change was made, but what the change does. Not how it was made, but what the change does. All right, so that's a pretty bell. No, that's okay. Sounds like my bike bell. I like to ring my bike bell for the very same tone. So maybe I'll pause before we go into what the wisdom from social scientists, and just give you a chance to ask questions or make comments.
that's what usually gets most people upset, is about, you know, what the change, you know, not how they made the change, but, yeah, how they made the change, you know?
Mm-hmm.
And you should, you know, just tell them that, you know, not to worry about that part, right?
Well, there's a way to deal with it, yeah. I hope it's useful. But yeah, that is the dilemma. I will tell you, you're going to, yeah, I put in a few concerns. Let me give you an example. One thing you should know, because everything I said sounded positive, and that's because I really do believe that the technology offers humans some really good tools. That doesn't mean every application would be something that I would be in favor of. So let me give you, let's think about an example. So what if, yeah, this might even be in the presentation, but I'll do it now. What if we have a genetic trait? That's a little bit too complex. I'll need to solve. Well, all right. So you know that there's been a lot of consolidation in the seed industry, right?
Yeah.
That's been driven in part by the revenues Monsanto has made by selling traits, right? And the traits are purchased typically, as I talk to producers. They buy the traits because they like what they do.
You get a patent on it, don't they?
They got a patent on it. They'll last for 20 years, and so they've made a lot of money, right or wrong. They've made a lot of money doing this, and they've used some of those revenues to sort of buy up smaller seed companies, and that consolidation of the seed industry. I have friends that are concerned about that. I would say my level of concern is probably a little lower than some of my friends, but I think it's a legitimate reason to be concerned about the technology. It's really not the technology's fault, but it's the use of the technology that has led to some of these consolidation trends. And so, that's an example of a concern that I think is legitimate. Okay, and I'll give you another. No, I'll give it to you right now, even though I don't have the slides for it in front of me. All right. So in 1970, you have to understand the history of southern corn leaf blight. In 1970, southern corn leaf blight, does that sound familiar to you? Yeah. That disease, even though we knew about it before, it was a disease that was known, it raced through the southern and midwestern corn belts and caused huge damage, way premature death of the plants, and attacked the ear, which was unheard of. What was going on? Well, it turns out, it was discovered over time what was going on. What happened was, first of all, another bit of background. There was a gene that was used. It was cytoplasmically inherited. It was maternally inherited. And there was a gene. They didn't understand the nature of the gene, but they knew that gene conferred male sterility. All right, so the corn that had that would not produce viable pollen. So why is that useful? Because if you're a seed company, you want hybrids to cross. You don't want your hybrid to self-pollinate. So you want that male sterility in the maternal line, and you want to plant pollinator rows next to it. This all sounds good. It's useful. It was very helpful. It was so successful. It was widely used in like 70% of the corn in the United States. Okay. So far, why does that matter? Because that gene, that cytoplasmically inherited gene, conferred hypersusceptibility to southern corn leaf blight, to a new strain of southern corn leaf blight. So far, I've not made a connection with genetic engineering, because that was all conventional breeding. So the danger came about through conventional breeding. But if you have a trait that you've carefully shepherded through the laboratory processes and through the regulatory processes, you're going to want to license that trait, and to as many seed companies as will buy it from you or license it. And so genetic engineering, I think it has a little bit more of a risk than conventional breeding in that area, because there's such an economic incentive to use your trait widely. Okay, so that's a legitimate risk. So that I've sort of steered into that direction, so you know that I'm not here to tell you that that all is perfect about the technology, but but that, but there are some issues, and we'll probably talk about some others. But the food safety question, again, I'm jumping around here, so apologies for that. But the food safety question doesn't really hold up to scientific analysis. And the publication again is that I'll refer you to is the one that says consumption of genetically engineered or GMO crops. And that's not just me talking, that's not just the National Academy of Sciences, which is the premier body of science in the United States. So the first quote is from the National Academy of Sciences. The second quote is from the National Academy of Sciences. Different years, different reports. They keep going back and reviewing the same thing and finding out the same answer. But the third one, take a look at that. Third quote. That's Europe. That's Europe's basically Academy of Sciences. So anyway, it kind of goes sort of flowed from your question. Yeah. Did that?
No, you did fine. No, the reason why I ask, I grow vegetables and I get in farmers markets. And people ask all the time, you know. Over in Europe, are they all tight about GMOs and all this other stuff too, or it's not?
Yeah. Europe is okay. So what's going on in Europe is the public. Their scientists are pretty much like ours. Have very common understandings, and you can read that. And you'll see that the quote that I've selected is representative of what they say. Their scientists even have done a lot of good work on genetic engineering. A lot. I read it. I've interviewed them for the podcast, and they're really good at genetic engineering. But their publics, France, Italy, Germany, they're very opposed to genetic engineering. So my feeling about that is I respect that that's where they're at, and they shouldn't have it forced on them. They don't want it. They shouldn't have it. But it's not for scientific reasons. Their scientists are on the same page as we are. Any other comments or questions?
With what we're seeing, they're talking about 10 or 12 GMOs.
Right, crops.
Of those, with the modifications that are made, are any of them have any effect at all on the edible portions?
Well, I mean, you'll have less insect injury on corn ears that have the.
Yeah. No, I guess.
You mean a negative?
Yeah.
No.
Actually, the comment about the damage to the corn ears would suggest that there is a positive health benefit if you don't have the fungi that are producing mycotoxins, which are incredibly acid.
Yes, and that's been demonstrated. So that's not, we're not speculating. That is absolutely.
Yeah, it's the same with your wheat and what you're talking about with the leaf mold. It still has no effect on the grain per se. What your modification made has nothing to do with grain source at all.
Right. So
anybody that's getting nervous about their GMO wheat, it's founded to start with.
Yeah, it's very unfounded because actually there is no wheat that's genetically engineered in the marketplace. A lot of people think there is. Yes. So let me think, what was some of the point you
I mean, with the way the weather patterns are and everything, you got to keep advancing. You can't keep planting the status quo. What was good 20 years ago, it ain't any good today. Like I've said, I've seen it in the vegetable seed. What my father planted and what I planted. What I planted is 1,000 times better.
Yeah, it's a big difference.
It's a big difference.
I saw a hand go up back here. Is there any question you have?
I kind of looked at the paper and handed out the answers to the questions.
Oh, okay.
But,
well, that's good.
Yeah. Labeling would, so obviously genetically modified pigs included, like corn or something like that. But I'm not selling fruits and vegetables, I'm selling animals that ate the food. So it's that I haven't looked into labeling. Do you know off the top of your head is that I'm not against labeling, but I'm just curious if that's something that happens at the label. So my turkey ate GMO feed, so we have to put on the turkey.
You know, that's a great question. I think I know the answer, but I'd rather refer you to the USDA when they start.
Well, what do you think the answer will be?
I think the answer is if you're feeding non-GMO feed, or GMO feed, that's not, the turkey is not engineered, so therefore
yes.
it's not bioengineered.
But it might not
People ask
be,
it,
it might not be organic.
But it might be organic.
Right.
Yeah, yeah.
Organic persistence.
Yeah.
That's
People are
what
trying to
it is.
like, are your turkeys genetically modified? I'm like, no, the turkeys are not genetically modified. Their feed is not genetically modified breed, breed.
No, I think that's what I expect.
No, I can clarify. So I participated in lobbying efforts towards the GMO labeling of the federal legislation, and milk and meat are exempt.
Oh,
So
okay.
your animals that consume GMO products does not need to be identified as GMO.
Is the labeling going to say GMO free, or it says yes GMOs, no GMOs, or is it just one of the other?
So let me, where are we? 31. So there's going to be, this is one of the ways that food producers can identify their genetically engineered products as genetically engineered, bioengineered is a phrase the Congress used in the law back in 2000. Was it 16? Yeah, I think it was 16. Yeah, yeah. Yeah, you're welcome. Okay. So, is there a question? No? Okay. So, guidance from social scientists. So, this is I put this in for the professional educators, but for growers and anybody, this is really good information. Actually, most of what is in here, in this part, is good if you're talking about anything controversial. One of the guidelines that surprises scientists very much is that presenting the science is necessary but not sufficient. It is not enough to present the science. And I'm looking right at this when I say this, because we're trained to present lots of information because that's what our department expects, right? Our academic department. But when you talk to the public, that is not enough. This has been shown over and over in the social science literature. So we'll talk about what you do say if the science. All right. You have to know the science. You have to be solid on the science. But it doesn't give you license to present anything you want. But you have to start with the science and then go further with your communication. I also like to tell people that anybody that talks about food systems issues and food production is there's going to be a distribution of reactions to what you say or what you do on the farm. And there are going to be some that really like it and some that are very opposed. And those are not the one. It might be different if you're trying to market it, right? So, but the approach I take for the general public is these extremes are not the ones that I don't want to get sucked into unproductive discussions with the extremes. It's the middle where I think this is probably a middle crowd. You come in with different opinions, but you're all here, and I can tell you're listening, and you're thinking, and you're offering your own thoughts. And that's the group that I like to talk to. Okay? All right, so I got to be honest here and tell you, I know what to do to promote dialogue. I do know what to do. I'm not always very good at remembering or implementing what I'm telling you to do. So I have to own that. I can tell you about some of my mistakes, all right? But I do know that there's a lot of good foundation for this, so I'm going to give you what is useful. I like this slide because whatever you do as a producer, people have a lot of respect for farmers. It's a pretty highly valued profession. So if you talk about your farm, people will listen. I mean, you might have each had some bad experiences. Remember, there's some distribution, but my experience is farmers carry a lot of weight, especially when they're talking about their own farm, their own family. Okay, this is because I work doing this stuff as part of my job, so I'll give talks like this, and normally this is the title slide for my talks. So when people come in, they see that, and they know right away, oh, we're going to have a conversation, it looks like. Well, at least have some back and forth. And so that's a good way to start. This is a way of implementing one of the good guidelines I've got for the social scientists is let the critics speak first. Whatever their criticism is, let them speak first. We're going to come back to this. Yeah. And here's where it gets hard. Right? If somebody, I mean, I don't know what your experiences have been. Probably most of them have been good, but maybe some have not. And it's, in moments like that you got to try to remember to take a breath and, and try to implement some of these practices. Okay, so resist that impulse to respond. All right, I think I've got more slides on this. I hope I do. Shoot. Because I got some good stuff. But anyway, we were talking about food safety. This is it's really good communication, very effective. It's totally not consistent with the scientific findings. Yeah. So, all right, this is so let me tell you about this. This one's kind of like a little story. I was on this field trip to look at cover crop systems in North Carolina. And this was the bus group, and I think it was one of the people, it was a friend of one of the people on the bus, and I were going through the lunch line together. We were standing next to each other, and she said, while we were waiting, she said, so what do you do? I said, oh, I work at the University of Kentucky. I do outreach on genetic engineering. And she gave me a look like that was, I thought, oh, wow, just, sorry about that.
Devil incarnate, huh?
What's that?
Devil incarnate?
Yeah. Yeah, but she was a sweetheart, and so she gave me a chance, and I said, so tell me, what is your principal concern? What is your number one concern, number 1 and 2? And she told me, and what did I do next? I asked her if it was, next thing I did is I repeated back what she said. Okay. And so then she knew she was being heard. And then the next thing I did was I said, would you be interested in what I've learned about this subject? And she gave me that same look again. But she said, yes, it's okay. And everybody has always said yes when I've asked it in that way. And so what did I do? First of all, I let the critic speak first. I let her speak first. As much as you know about your farm, if you can let them speak first, the tension's going to go down, even if you don't like what you hear. Even if it's not fair, practice active listening. Repeat back what they say so they know they've been heard. Okay? If you do these 2 things, the tension's going to be lower. And then third is, if you ask for permission, right? Seek consent. Ask for permission to share. They're probably going to say yes. Okay, so these are some of the principles that have been helpful. All right, this one is more for the scientists in the room.
Man, I can't read it.
Yeah. Yeah, exactly. This is kind of what we're doing up there sometimes. So, so the point here is don't bombard the public with everything we know. And that probably could apply for producers as well. But certainly we do that. We go way overboard on that as scientists because we're trained to present lots of data, and that's not how you approach a controversial subject with the public. All right, so this was a news conference I gave in Bolivia, and I'd never given one before, but it was kind of neat, with the cameras and the lights and the microphones and everything. And so the last question, I'll never forget it. The female reporter who asked, she said to the minister next to me, who was Minister of Agriculture for the province, she turned to him and said, "But aren't these dangerous to eat?" And he started to answer, and I thought, what can I say in 30 seconds? Because that's all I'm going to have. What can I say to address this question in Spanish? And it was really good because the minister talked for a minute or two, and he gave a good answer, but mostly he gave me time, and I knew what to say. And so when he was done, I turned to him and said, "Thank you, Minister, that was very good. Would you mind if I add to it?" And he was like, "Of course, please." And so I turned to the reporter and I said, and I meant it, and I said it from the bottom of my heart, I said, "We scientists want safe food too. We scientists want safe food too. For ourselves, for our families, and for you and your family." And it's true. I think, is every scientist totally integrous? Well, I think most are. Most are. As good of people as anybody, any group we encounter. And I think we do want safe food. We don't want to go to bed thinking we could be causing harm to the American people. So what I was doing there, to put it in the language that a social scientist might do, is I was speaking to shared values. And the shared value is safe food. We already talked about farmers. It helps, when you speak about your farm, it helps personalize the survey and humanizing technology. So does this. CRISPR technologies are being used by graduate students worldwide, especially in the countries with developed economies, just everywhere. And that helps showing pictures of the people doing it helps humanize the technology. Okay, so this is for all of us here. I've been there. It's, when you feel like you're being treated unfairly, that's a very common and human reaction. But it's very important to keep it respectful. And and so let me tell you about an example of my own where. So one of the largest environmental organizations in the country had an article that was incorrect. There was an error in it. And so I wrote to them, but I talked to my wife first, and she said, "Well, here's some guidelines for you." And I wrote to them, and I said, "Thank you for the work you do, the important environmental work. It's very, I know that some of the very important accomplishments you made," which is all true because Judy's a member, my wife is a member. And I said, "Now that on this particular topic, your article has an error, and here's the error." And this is what they wrote back. I didn't know what I was going to get back, but she wrote, "Dear Dr. Vincelli, thanks so much for contacting me. The biotech team agrees with what you wrote." They're still, their position in the organization is still anti-GMO, and that's their choice. But we had a good exchange, and that was because I kept it respectful. And another thing I'll recommend is thanks for your feedback. I've done this several times on the internet. Somebody writes something to me that's, it's sort of inflammatory, maybe a little bit of some germ of truth, but maybe some inaccuracies, any of those things. I've done this. I don't have to respond, or I don't have to respond in kind. I can just say thanks for your feedback. And I do that, and the 2 times I've done it, I've gotten a little smiley face back or something like that. So that's a way to diffuse, and if they come back angry again, you just block them and tell them that you've been unprofessional and unkind, and I'm blocking you. But I've had to do that very few times. Empathize. I love this this statement here. People must know how much you care before they care how much you know. Yeah, that's good. So we talked about the consolidation of the seed industry as an outcome of at least some of the use of genetic engineering. So Monsanto, Syngenta, DuPont. This is all as of 2009. Yeah, sorry. Yes, it does, because Monsanto has been bought out by Bayer.
Yeah.
Right,
I
and
can't believe that the one bought the other, because the little red dot ate the large red dot.
yeah, that's that. I think this is just.
Oh.
What is this? That doesn't represent their whole. It's just a seed, and I think it's their seed based on seed sales or something. So it's not their
Right.
revenues. Yeah, that's why Monsanto's so big compared to Bayer. Yeah. You know, so there's been a lot of consolidation. And you know, it's a legitimate concern, and I gain credibility when I say that. I think I speak honestly, but I do think it's a legitimate concern. But you know, but it also gives credibility to me. Okay, so this is a topic that yes, sir.
And your concern there is mostly due to the lack of diversity.
Yeah, well, I'll tell you what I understand, and then others can offer some opinions too. I think the concern is that it puts too much power in the hands of, you know, a small number of companies for the seed supply. So does that capture the concern that others might have?
I always think monopoly is never good for the consumer. And if you're a farmer, consumer seed.
Well, I'm looking at more from strictly the ag in the field side of things. You know, if Monsanto, you know, for a name, is producing 90% of the seed, it's all going to be the same.
Mm-hmm.
You know,
yes, that could be true.
Whatever.
Yeah.
So we're going to lose that diversity to make our
You're going to lose the edge of competition too.
Yeah, no, I understand. That's the economic difference.
Exactly.
Both of those, I think, are
very
important. Yeah.
Is there quite a few, like, you know, small family seed farms still out there, or not? Or not? They
I don't
kind
know
of going
about
by
seed
the
farms,
wayside.
but seed companies.
I mean, seed companies.
Yeah, there are a couple companies that are startups that are using genetic engineering, you know, outside of the Monsanto behemoth. But Calyx is one. Can't think of the other, but I'm sorry. The question was
if there was like
small seed companies.
Yeah, more independent. Yeah,
there's still a few out there, but there's been a lot of consolidation. Yeah. And it's only part has been driven by genetic engineering, but most of Monsanto's purchases probably were driven by the revenues of genetic engineering. So, you know, I always point that out. Yep.
The reality is that me, the farmer, is not going to Monsanto for my seed. I'm going to who our local supplier is, and our supplier is going to, the choice is dictated, not necessarily, I know what traits I'm looking for, and he knows what traits I'm looking for, and so the reality is what's most readily available is going to be, you know, so when it comes to shopping for seed,
like the large companies.
Yeah, it is, you have to stop using certain agencies and ag service providers if you want to go find that small family farm seed supplier.
So they may exist, but there's a lot of sort of hurdles into actually tapping into their products.
I think so.
Okay, sure, that could be it's good addition.
There's one practice I find extremely disturbing, and that is the dicamba herbicide resistance trait unfairly penalizes neighboring farmers that don't have the trait and forces them into getting that trait. And that is something that, in terms of antitrust, I think ought to be pursued.
Right.
Because it's completely wrong that EPA allowed a volatile herbicide genetic resistance to be engineered into corn and soybeans.
Okay. Yeah.
From an environmental point of view, it was a terrible mistake of EPA to allow that registration.
So let me follow up on that. So we're talking about part of the way that that the ag industry is dealing with Roundup Ready resistant weeds. So does everybody understand the concept of Roundup Ready? It's been engineered to be resistant to Roundup herbicide. This is what I say about it, and then I want to follow up to your point. Roundup Ready crops are useful but not sustainable. Useful but not sustainable. They're useful, and I always say if you don't know why they're useful, talk to a farmer who uses the technology, and then you'll hear why they're useful. Not sustainable because what happens when you use the same herbicide active ingredient over and over?
Weeds build up, certain weeds will build up resistance.
Yep. And that was not expected by Monsanto, I understand, back 20 years ago or 25. But it happened, and so useful but not sustainable. And I've never had a farmer dispute that, by the way. So that's on the pesticide treadmill. And then the element that, I'm sorry, what's your
Rich calls.
Rich has brought in is now, because of this pesticide treadmill and the resistant weeds, we're going to add dicamba herbicide and engineer resistance in soybeans to dicamba herbicides. We can kill out the resistant weeds, the resistant weeds that are resistant to glyphosate. But now we're requiring the use of both of these active ingredients, glyphosate and dicamba. Oh, and the dicamba drifts and can damage your neighbor's fields unless they use dicamba-resistant beans through the technology of Monsanto. So this is a mess, okay? And we can talk more about it, but here's the role it plays in communication. Let's be honest. Let's talk about the weaknesses of the technology, because it does help build our credibility. And frankly, if that's what we think, then we owe that right to our constituents, our neighbors.
I guess I really have a hard time. I had a hard time last night, and I'm glad someone did bring it up. It's all talking about the plant pathology. You're never talking about the soil. And I guess, I mean, you use Roundup in your driveway. I mean, it kills.
Sure.
So you're just, depleting the soil structure. And that's what feeds everything. Right. That's the foundation. That's the basis.
Yeah.
So I guess that's, I guess I just really always have a hard time.
Right.
Because you're just, not talking about the soil,
Yeah, that's a good point. Yeah, I think I heard some feedback afterwards that there was some dismay that hadn't been covered. And I guess I do want to say, we had 15 minutes to talk and a lot of questions. So that'd be the only reason it didn't come up in the questioning. So that's why it didn't get covered. But I was thinking what would be my answer, and what tonight you're here. So it might have been you, it might have been somebody else, a colleague overheard. But I don't know of anything about the technology of Roundup Ready or BT that would be harmful to the soil structure or the soil microbiome. I think that those things are not only important, they're real, but they're driven by things like the organic matter introductions into the soil, the farming approach, and so on, but Roundup could be used in ways that probably are destructive to the soil. And Roundup could be used in ways that are beneficial to the soil, especially if it's used to promote no-till farming, which can increase the organic matter of the soil and improve the soil structure. So I've never seen any connection in the scientific literature between soil health and the genetic technology. It's maybe the farming practices can be questioned, but that's the farming practices, not the genetic management of the trait. So, yeah, that's a good question, but I don't think there's a connection. And I guess I'd ask you, if you read through probably the single most important one to look at, 600 pages, is the second one on the list. You can skim it pretty easily, even though it's 600 pages. If you find out that there is a soil health connection with genetic engineering, I'd like to read that. So you feel free to contact any of these
extension people, and they'll get a three-way going. So.
There is another rather disturbing thing about the Roundup Ready and herbicide-resistant large commodity crops, and that is how coercive they are on a macroeconomic scale.
Mm-hmm.
I have a friend who lives in Mansfield, but she has a farm in the Dakotas, and she came to me asking whether I could help her figure out some sort of an alternative to using Roundup Ready crops. And so I asked her what the return per acre would be for corn or soybeans. And there would be absolutely no way, with the overproduction of those crops and the very slim margin, to be substituting selective alternative herbicides and still make a profit.
So the
only way
okay.
you might be able to make a profit is if you start out completely dependent on using the Roundup Ready crop.
Because the economics favor it over the other.
Absolutely. Absolutely. So the macroeconomics are such that there's such an oversupply, partly because of the Roundup Ready crop, that the only way you can maybe turn a profit is if you continue on that track.
You know, that's a really good insight. That reminds me of what Don, I'll think of his name later, highly respected grains farmer in Kentucky. He passed away just a year or two ago, but really extremely well regarded. And so I asked him when I started doing outreach on genetic engineering. I said, "So Don, what do you think about Monsanto?" And he paused and he said, "Well, I like the traits. I don't like Monsanto." Yeah.
Mm-hmm.
So, never forgot that. We can loop back to these. These are on the question of safety of glyphosate, or the lack of safety of glyphosate. So if you're interested in that topic, we can loop back, but I want to make sure we get through.
Can you go back to the last slide?
This one?
Yeah.
Yeah. I can explain that. It just was published, as you can see. Yeah. Yeah. Yeah, that was my reaction when I saw this paper.
Wow.
Yeah. So, all right, since you're studying it, I'm going to give you a little explanation of what it means.
Yeah.
Yeah.
You can go through.
Yeah. Yeah. I didn't put in slides to show the data, but what they did was they worked with rats, laboratory rats, and so the mother is generation 0. The fetus that she gives birth to, the mother was exposed to Roundup. The fetus, generation 1, was exposed to Roundup in utero, okay, while she was in the mother's uterus. The generation 1 rabbit that had been born gave rise to a generation 2, and the generation 2, the cells that give rise to this animal's gonads was in this animal when it was exposed in utero. So that's generation 2. And then generation 3 would have been the generation born from this animal. Generation 3 had no exposure anywhere, anyhow, to Roundup. And what I remember from looking at the data charts was that there were more negative impacts in some of the later generation animals than in the mother. And and generation 3 I think had the most. Generation 4 had quite a few. Generation 2 had quite a few. Generation 3, remember, no cell was ever exposed to Roundup. But there was a number of notable health effects. So now they used I'm not a toxicologist, but I thought the dose could be argued to be pretty high, 50 times what would be considered the safe level by EPA. But we can't blow this off. You know, we can't blow it off. It's a legitimate concern. And I will say, I try to study how pesticides are regulated pretty thoroughly. And this idea of testing the generational toxicology, you know, the exposure in the mother, and then, you know, all the way to her grandchildren, great-grandchildren, sorry. Mother got exposed, children, grandchildren, great-grandchildren are all assessed. And that's what generational toxicology is. That's not a routine part of pesticide evaluation. So,
but these sorts of effects have been seen, for example, the fossil and the fox.
Yes.
Both have epigenetic.
Yes, exactly. Yes, you're right. So, I think one could argue that we need glyphosate aside. I mean, not put that aside, but this is a bigger issue than glyphosate. You know, maybe we need to be assessing pesticides in this way as well. I think there's enough data, because I've seen it within causal data, that we should actually do that.
So was this a study or just a review?
No, it was a published experimental study. Yep.
So the date on that looks like it was this week.
Yeah.
Wow.
That's about right. Yeah. Hey, you know how I found out about this? Twitter.
Oh, wow.
If you follow the right people, you got to follow the right people. If you follow somebody that says, I ate a cookie, that's what you'll get. But I follow scientists and journals and things like that. And I found out about this on Twitter. So I actually read it this morning. Was it this morning? Yeah, it was this morning. Yeah, because or 2 days ago. But it was something I said, I got to make sure I put this in because it's relevant. Yeah, sure.
So, in terms of pathology and education, so is that suggesting that glyphosate is a mutagen rather than mutagen?
No, I don't.
I could jump in. Epigenetic effects are usually indirectly affecting how genes are expressed, but not in the individual exposed, but potentially in their offspring or grand offspring or further along down the road. There's an amazing human study, I think it was Sweden, from a case of a famine that took place. And quite remarkable epigenetic effects resulted in either children or grandchildren having a high incidence of diabetes from the parents having been exposed to famine at the time that their gonads were developing. So the grandchildren then had diabetes, and that was an epigenetic effect which would be in parallel with the kinds of changes that are seen here. So it's not mutagenic, it's how the genes are expressed.
Right. Yeah, let me add to that. It's right. It didn't actually change the DNA's, the sequence of the DNA. It changed how the DNA was expressed. Okay. And so that would usually, as I understand it, usually epigenetic effects beyond a certain number of generations disappear. Mm-hmm. And so these are not. I'm not dismissing the concern. These are not indefinite permanent changes in that population's germline, but it's enough, there's enough data there for me to say, whoa, you got to pay attention. Yeah. Okay, so I do have a few examples of applications that are above and beyond what you've heard. I expect, so I'd like to give those to audiences as well. First of all. You brought this up, aflatoxin and other mycotoxins produced by fungi. They're natural and they're usually quite dangerous. Aflatoxin is, we see it sometimes in Kentucky corn. It's a very potent liver carcinogen, very, very damaging, very damaging. So here's an example of an application that really I thought, was exciting as far as the use of genetic engineering. This is experimental, so it's not being used in the field, but it really it sort of took my breath away when I saw it, so I like to describe it. So they inoculated with the fungus in these parts right here. You can see a little bit of the fuzziness of the spoliating fungus. And they probably did that around the blister stage or so, somewhere around there. And then a few weeks later, they sampled the kernels surrounding the inoculated site, and they sampled for aflatoxin. These three bars are the controls that they used in this experiment, so there are no transformants, and that's a lot of aflatoxin. That is huge, dangerous, bad, and that's what you want in your experiment. You want to see effects like that. And these are 5 of the gene silenced. Yeah, that was, I think, scientists all over the world had the same reaction because I had never seen a study that shut down high levels of aflatoxin too soon. Now, it's not been done in the field. This is a growth sample test. So we don't know if it will really function under field conditions, in grain that's approaching maturity and things like that. But, well, how so?
If it works.
Oh, yeah, if it works.
It's if you can eliminate aflatoxins in the dairy herd, you're looking at 15 pounds of milk per cow.
Wow.
Okay. Boy, that's really what I do.
That's incredible.
Yeah. Well,
it can be that, what you're showing for the liver problems with them, you can get into digestive problems and feed intake and everything else on the real life.
Yeah. Okay. Well, this is the population I thought of right here when I read this. In developing nations with developing economies, they get exposed much more than we do to aflatoxin and other mycotoxins. So, it would be their choice to use these technologies, but that was pretty impressive result. So this is I show this because it speaks to shared values. Our shared value might be something like safe food for those of limited means, or for everybody for that matter. This is BT Brinjal. You've heard of this?
Sure.
Yeah. So in Bangladesh, Brinjal is eggplant, and it's a very important crop culturally, and it's been engineered for resistance to stem borer, and it's been very popular among farmers. And that slide kind of says it all. The farmers that use it have dramatically reduced their insecticide use. So that's a good example of why the technology might have a place for some applications. I'm going to probably see this disease next month when I travel to Africa. And this disease, it's a viral disease, causes very severe damage to their cassava. It can even be lethal to it. And if you harvest anything at all, it's going to have these necrotic spots in it. And so this is one of the African farmers. So this comes from African farmers, not farmers, African scientists working on African problems to come up with African solutions. And there are others that
that's possibly related to like the papaya ring spot story in Hawaii.
It's very similar.
Now, gene silencing through expression of the virus protein code or something like that.
Yes, exactly right.
So the question is, if you eat the cassava or the papaya that's engineered for resistance to the virus, would you be eating any more of the DNA or RNA of the virus from the genetically engineered crop than you would if you ate a viral infected fruit?
Right. I've never done the calculations, but every bite you take out of the engineered fruit is going to have protein from the virus, right? That's the mechanism. It's like a vaccination in that sense. But the level but with the infected fruits, right, and non-engineered ones, you're eating the whole virus.
Yeah.
It's not just the protein. So, and you're eating. I've been told a lot more, but I've never done the calculations. Either way, it's the virus doesn't affect humans. So we've been eating it for thousands of years, I guess. And so I guess my point to close is case by case. I think that's my thinking about these, but that's just me. So actually we've done very well. I think we've left time for continued discussion if there's more questions or anything.
follow up on his point about, like, you're consuming the virus either way. And your response is, well, the virus doesn't affect humans.
Right.
So when it comes to communicating with people, like, if you're at a farmer's market or somebody's grabbing the sweet corn from, you know, a bushel basket, like, you've got a very finite amount of time. Like, and I appreciate the importance of listening to the critic, but I'm wondering about, like, there's the propaganda component of, like, this really scary, terrible thing, but I feel like I almost want the propaganda on the other side of it to say, like, well, you're eating a virus either way. You know, like, I don't know, I'm simplifying it a lot, but recognizing that I've got to have an elevator speech.
Right.
And I need it to be good communication, like the scary poster. But I want to be legitimate. I want to be factual.
Yeah.
And so I just kind of grabbed on that because I thought that was really interesting. That, like, that is, like, very intriguing thing to think about, how you're taking that virus, regardless of if it's the genetic engineered version or not. But then in the end, it doesn't even matter.
Yeah, especially because it takes a while to explain that. Yeah, I think I understand your point. And so I'm going to open that up for comments because I think I've been able to find ways to make it comprehensible, but now you know, I'm kind of stuck. I don't know how to make some of these really, you know, concise explanations the way you're asking for. So, but you guys might have better ideas than I do about that.
I would think one of the main things you got to do is figure out what plants you're talking about, which crops you're talking about that can be modified, and what those modifications are. You know, if it's for root boards that's got nothing at all to do with the grain, you know, that's an easy one to explain. You know, if it's, you know, something else that's got more something to do with the consumable parts thereof, it's a different story.
Sure.
And that's one of the things I was hoping to get more of a list of, you know, feed-wise, because it's the same thing with the livestock industry. You know, you get somebody to come into the farm, into the store, I've got to have non-GMO feed because it's healthier for my chicken. Okay. And, you know, and if that's not suitable for them, then I need to have organic feed for them. Okay? And it's real easy. You find out how convinced they are on what they're doing when you say this one costs $4 more and this one costs $8 more. I'll take the bag of regular feed. Yeah, where's your convictions at?
Are you asking, do you grow any BT sweet corn? Okay. So that would be sort of a case study for you, where you'd like to be able to address questions quickly, right?
Yeah.
Well, one of the facts I'll give you, I've done this calculation so many times. I've had my graduate students check it because it sounds unbelievable. But I took one of the most common BT traits in corn and did the calculations, you know, based on the LV50, if you know what that is, and you'd have to eat like 400,000 kilograms of corn to reach the LV50. The LV50 is the dose that kills half of the population. So it's actually, the BT protein is remarkably safe. And you have to eat 400,000 kilograms to have a 50% chance of dying.
Okay.
I mean, 400,000.
If you condense all of BT from that,
yeah, but it's
just, you can't physically
you can't physically eat that. I mean, it's like enough to fill
It's a good contest, though. Cool, okay.
I hope it's.
Yeah, it is, thank you.
What else?
Something big that I took away was that I might bring up to my customers when I'm having this conversation is the type of conventional breeding or ways, you know, to get reprocessing mutagens, things like that. Well, there's some pretty scary ways that they're editing genes, you know, not, well, that's not editing genes, but, you know there's scary ways to do it, not genetically edited.
Yes,
you know?
that's exactly right.
So it doesn't mean that your food's any safer than my food, you know what I mean, just my define as genetically engineered.
Any
That's just gonna scare them and they won't eat anything.
In any conventional breeding, you know, you've got your chances of bringing that many more or less to valuable traits than you do on engineering.
Because that's the thing, it's unnatural, unsafe, but mutagens are not natural. Like, or, I mean, it's natural, but the, you know, that's not something that's naturally happening either, just like cutting a gene with the, you know.
This has been going on for a while. Hybrid corn was developed in New Haven, Connecticut at the experiment station.
I didn't know that.
Years and years ago. I don't know what year it was, I forget, but.
About 1912.
Yeah.
Yeah, somewhere,
Well,
it was dihybrid crossed by Donald F. Jones.
Yeah. So that's
somewhere
where,
around 1920.
that's where it started. And then this year, I'm planting, usually I plant a bunch of yellow and green squash. Well, the green squash there's this one variety called Spineless Beauty. And the big deal with that is on the stems, there's no spine. So when you pick them, you don't get all cut up and scratched up. Well, this year I'm planting, I don't know, what's the name? It's a yellow squash, but anyways, no spines on that stem either. So I'm trying, I'm looking forward to that. So, you know,
It's
I
all
mean,
based on genetics.
yeah, it's all based on genetics. And the sweet corn of today is 1,000 times better than the sweet corn of yesteryear. You know, they had one that was, it was called Harmony. Oh yeah, it would produce like crazy, but 2 days later, oh my God, you better eat that tub over there. It tasted better. But today the hybrids, you know, they got today the SEs and the super sweets and stuff. It's you can keep them for 4 or 5 days and they're just as sweet. So, you know, there's big, you know, there's big improvements. I don't know. I feel that, you know, agriculture gets a bad rap from the press. It's just, I feel that we're easy prey, and you know, and the average consumer doesn't understand anything about it at all. The food's just there in the grocery store. It's just, you know, it's magically there every week. So
it makes me wonder how to bring up scientific literacy, because, you know, I'm aware of aspects that would just, I find it absolutely mind-bogglingly incredible that maybe you would have heard about it, that nobody else would. What percentage of our genes have resulted from previous generations of what's called horizontal gene transfer? Genes coming from viruses or from bacteria, but not from parents or grandparents or so forth. So the percentage of our genome is like 7% or more is from horizontal gene transfer, which is exactly the same sort of process as we're doing with genetic engineering. So when somebody says it's unnatural to be transferring genes from somewhere else into a plant or an animal, and it's an unnatural process, I say, no, you just don't understand biology. Because genes have to have had an origin from somewhere, and an amazing proportion of genes had an origin not directly from an ancestor, but from horizontal gene transfer, which is so, it's just incredible.
how does that work? Eggs and sperm, how does it get in there all the time?
Yeah, actually, the concept that Rich, did you say?
Yeah.
Brought up was what got me into doing this outreach. I started reading about horizontal gene transfer happening in nature. Now it doesn't happen like on short time frames over evolutionary time scales, but it's happened
Hundreds of years.
millions. But it's happened abundantly, and I think it surprised scientists when we first started discovering it. But it's so heavily documented. It's called horizontal gene transfer. In fact, my understanding is that the most common gene in humans, or genetic sequence, is from a jumping gene.
Oh.
Did you know that? It's ALU1.
Oh, ALU, yeah.
So we don't know how, you asked how it happened. It's a great question. We don't know how it happens in nature. It's sometimes associated with, it's sometimes thought to have been as a result of an infection. And maybe during the DNA duplication process in the cell, it made a mistake and included a portion of the virus as it replicated the DNA. We just know we see the genetic outcome. We see the sort of the baggage that was created from these horizontal gene transfer events. Some of them are functional in humans and other animals. They're actually they're not just residue genetics. They're actually functional. But we don't know how it happened, and probably never will. You know, we might have some clues in some cases, but it's happened so long ago that. Yeah, but that was it was reading about horizontal gene transfer. I was like, what? This happened in nature? You know, and that was what got me into doing this outreach. Yeah.
So what are you most excited about? What's on the horizon for genetic engineering that you think is like going to be really, really powerful, really exciting?
Yeah, that's a good one. The thing that most impresses me is the wealth of opportunity. There are public databases of genetics that are massive, that you cannot believe the information we know about genetics, and more, accelerating more every year. So I think, and here's the point I didn't make, but actually, when we do genetic engineering, we're using techniques we learned from nature, actually. We're using natural enzymes and things like that. So we're not trying to be nature, but we're using things we learned from nature. And so I would say the thing that impresses me most is the wealth of opportunity. It's true, you made the point about a lot of focus on pesticides, and that's probably because of my own bias. Well, that's not this, folks. But my own bias is focused on pesticides. I've done a lot of work with pesticides. And have tried very hard to reduce pesticide use, and it's been pretty frustrating. Except I think genetics offers an out for diseases and insects. Right? That's right. You're the
absolutely.
Yeah.
And like parasitic nematodes, you just go on and on.
Yeah.
From my perspective, I work at the Ag Experiment Station, and I have a Christmas tree. So, yesterday or day before, my department head says to me, oh, by the way, EPA is undoubtedly going to be pulling the registration of chlorothalonil fungicide.
Wow.
As a Christmas tree grower, I'm thinking, okay, we have needle cast diseases in Douglas fir and spruce, and chlorothalonil is the go-to fungicide for managing those diseases. We practically were several years behind in looking for alternatives, and we're going to have that particular tool removed. When I was in graduate school, I was in the cohort, the first group of students that received fellowship in sustainable agriculture. And I've always felt that being able to use sunlight in a plant to have an alternative to applying a pesticide is a better way to go. And that's what genetic engineering could do, is have the plant grow its own defenses that we can engineer into that plant so we're not dependent upon Monsanto, Bayer, whatever companies they are now, to buy products that we're spraying on. And that's where I think the power of genetic engineering will be in the future. And we're going to be forced to go that way as agricultural scientists, because eventually people find unacceptable risks in any of the pesticides practically that any of the synthetic products, not products that we use, but the go-to broad spectrum fungicides, herbicides, the handwriting is on the wall. We have to find genetic alternatives.
Yeah, I don't know.
Even the yield variable, that seems endless. You know, don't they, one time, 200 bushel corn, that was a big deal. Now that's the norm. Now there's guys out there that are striving for 350, 400 bushel corn, They say, my ag channel says 9 billion living on this planet by 2050, they say. Somebody's gonna feed them.
Yeah, that's the estimate. Well, I've enjoyed this group. You're full of a lot of good ideas, and thank you all.
Thank you.
Oh, yeah.
Thank you.
Yes, sir.
Thank you for coming, everyone, and safe travels home, and hope you enjoyed the presentation. It was useful. It was useful.